If we could we step back to the big, profound, philosophical, both tragic and beautiful question about age? How and why do we age? Is it from an engineering perspective? You said you like the biological machine. Is that a feature or a bug of the biological machine?
It is both a bug and a feature. Evolutionary speaking, we only live as long as we need to replace ourselves efficiently. If you're a mouse, you're only going to live 2.5 years, 3 years. You're probably going to die of starvation, predation, freezing in the winter. So they divert most of their resources to reproducing rapidly, but they don't put a lot of energy into preserving their soma, which is their body. Conversely, a baleen type of whale, a bowhead whale in particular, will live hundreds of years because they're at the top of the food chain and they can live as long as they want. So they breed slowly and build a body that lasts. We're somewhere in between because we've really only just come out of the savannas where we could be picked off by a cat. We were pretty wimpy going back 6 million years ago. So we actually need to evolve quicker than evolution will. And that's why we can use our oversized brains and intuition to give us what evolution not only didn't give us, but took away from us. We're pathetic. Look at our bodies. Mm-hmm. These arms, if any of us, even the strongest person in the world, went in a cage with a chimpanzee, the chimp could knock that person's head off, no question. Yeah. So we're pathetic. So we need to engineer ourselves to be healthier and longer lived. So getting to aging, we can do better, right? Whales do way better. We're trying to learn how whales do that. And if you ask really anybody in the field now, Professor, they'll say there are 8 or 9 hallmarks of aging, which are really, it's a word for causes of aging. So that you probably have heard of some of these, your listeners will have. Loss of telomeres, the ends of the chromosomes, like the little ends of shoelaces, that kind of thing. They get too short, cells stop dividing, become senescent. They become, they put out what are called mitogens that cause cancer and inflammatory molecules. So that's another aspect of aging, cellular senescence. Another one is loss of the energetics, so mitochondria, the battery packs, wind down. There's a whole bunch, stem cells, proteostasis. Well, these are our Achilles heels that I'm talking about, that are common amongst all life forms, really. But if you want me to jump to the chase as to what is the upstream defining factor, if we boil it down, what do we get? So most biologists would say you can't boil it down. It's too complex. I would say you can boil it down to an equation, which is the preservation of information and loss due to entropy, i.e. noise. And that is the basis of my research. It originally came out of discoveries in yeast cells, where I went to MIT in the 1990s.
You studied bread.
I kind of did. I studied the makers of bread, a little yeast called Saccharomyces cerevisiae, which at the time was one of the hottest, excuse the pun, organisms to work on.
Yes. But
we figured out in the lab why yeast cells get old and found genes that control that process and made them live longer, which was an amazing 4 years of my life. One of those genes had a name with an acronym SIR2. Now, the 2 is irrelevant. The SIR is important, and the most important letter out of all of those 3 is I, which stands for information. Silent information regulator number 2. When you put more copies of that gene in, just put in one more copy, the yeast cells live 30% longer and suppress the cause of aging, which was the dysregulation of information in the cell. And then, so fast forward to now, I've been looking in humans and mice, because they live shorter and are cheaper to study, whether loss of information in our bodies is a root cause of aging. And I think it is.
Your boldness in viewing biology in this way is fascinating, because that also leads to a kind of, it's almost like allows for a theory of aging, like you could boil it down to a single equation, and it leads to perhaps a metric that allows you to optimize aging, sort of in the fight against entropy, to figure out which mechanisms, like you said, the silent information regulator, which mechanisms allow you to preserve information without injecting noise, without creating entropy, without creating degradation of that information. For some reason, converting biology, which I thought was mostly impossible, into an engineering problem feels like it makes it amenable to optimization, to solving problems, to creating technology that can, whether that's genetic engineering or AI, it makes it possible to create the technology that would improve the degradation of information and aging. Is there more concrete ways you think about the kind of information we want to preserve? And also, is there good ideas about regulators of that information, about ways to prevent the distortion, the degradation of that information?
Right. So we have silent information regulator genes in our bodies. We have 7 of them, SIRT1 through 7 they're called. And we found in mice, one way to slow down the loss of information is to just give more of these, to upregulate these genes. Mm-hmm. So we made a mouse that has more of this SIRT1 gene, turned it on, and that slowed down the aging of the brain and preserved their information. Now, what information am I talking about, you might ask? Well, again, you can simplify biology. There are 2 types of information in the cell primarily. The one we all read about and know about is the DNA, the genome, and that's base 4 information, A, T, C, G, the 4 chemicals that make up the various sequences of the genome, billions of letters. And that also degrades over time. But what's been fascinating is that we find that information is pretty much intact in old animals and people. You can clone a dog. One of my friends in LA just cloned his dog 3 times. So this is doable, right? It means that the genome can be intact. But what's the other type of information? It's the epigenome, the regulators of the genetic information. And physically, that's really just how the DNA is wrapped up or looped out for the cell to access it and read it. So it's similar to, and excuse this analogy, but it's a good one, a compact disc or a DVD. Those pits in the foil are the digital information, that's the genome. And the epigenome is the reader of that information. And in a different cell, you read different music, different songs, different symphonies. And that's what gets laid down when we're in the womb, and that makes a skin cell forever a skin cell and not a brain cell tomorrow. Thank God. Otherwise, our brains wouldn't work very well. But over time, what we see is that the brain cells start to look more like skin cells, and the kidney cells start to look more like liver cells. And they, what we call X-differentiate. This is a term that we use in my lab, but isn't yet widely used. But we needed a term to explain this. And that process of X-differentiation, the loss of the reader of the CD or the DVD, we liken that to scratches on the DVD so that the reader cannot fully access the information. Now, we can slow down the scratches, as I mentioned. We can turn on these genes. We can even put in molecules into the cell or even eat them and turn on those pathways, which my father and I have been trying to do for about a decade to slow things down. But the question that I've had is there a repository of information still in the body? Because anyone who knows anything about the loss of information or even has tried to copy a cassette tape or photocopy or Xerox anything knows that over time you lose that information irreparably. So I've been looking for a backup copy, inspired largely by Claude Shannon's work at MIT as well in the 1940s. His mathematical theory of communication is just brilliant. And so I've been looking for what he called the observer, which is the backup copy. We today might call that the TCP/IP protocol of the internet that stores information in case it doesn't make it to your computer. It will fill in the gaps. And we've been spending about the last 5 years to try and find if there really is a backup copy in the body to reset the epigenome and polish those scratches away.
That's incredible. So finding the backup, so whenever there are too many scratches pile up, you can just write a new version. Like write not a new version, but go to the backup and restore it.
Right. That's really all we're talking about. It's not that hard once you know the trick.
And for people that actually remember like DVDs and scratches on them, how frustrating it is. That's a brilliant metaphor for aging. And then the reader is the thing that skips, and then it could destroy your experience, the richness of the experience that is listening to your favorite song.
Right, but in biology, it's even worse because you'll lose your memory, your kidneys will fail, you'll get diabetes, your heart will fail, and we call that aging and age-related diseases. So most people forget that diseases that we get when we get old are 80 to 90% caused by aging. And we've been trying to fix things with Band-Aids after they occur without even generally talking about the root cause of the problem.
Is there the scratches? Do those come from? Are those programmed or are they failures? Meaning, is it so if it's by design, then there's like a encoded timeline schedule that the body's just on purpose degrading the whole thing, and then there's the wear and tear of like the scratches in a disc that happen through time. Which one is it that's the source of aging?
It's more akin to wear and tear. There isn't a program. Getting back to evolution, there's no selection for aging. We're not designed to age. We just live as long as we need to, and then we're at the whim of entropy, basically. Second law of thermodynamics, stuff falls apart. We live a bit longer than age 40, only because there are robust, resilient systems, but eventually they fail as well. Current limit to the human lifespan where they completely fail is 122. But I don't like to think of it as wear and tear because there's two aspects to it. There's a system that's built to keep us alive when we're young, but actually comes back to bite us as we get older. And we call this issue antagonistic pleiotropy. What's good for you when you're young can cause problems when you're older. So we've been looking what is the main causes of the noise, and we found two of them definitively. The first one is broken chromosomes. When a chromosome breaks, the cell has to panic because that's either going to cause a cancer or kill the cell. There's only two outcomes. It's pretty much a problem. And so what the cell does is it reorganizes the epigenome in a massive way. What that leads to is, think of it as a tennis match or a ping pong game. The proteins are the balls, and they now leave where they should be, which is regulating the genes that make the cell type, whatever it is. And they have a dual function. They actually go to the break, the chromosomal break, and fix that. And then they come back. You might ask, well, why is it set up that way? Well, it's a beautiful system. It coordinates gene expression, the control systems, with the repair. You want them coordinated. Problem is, as we get older, this ping pong game, some of the balls get lost. They don't come back to where they originally started. And that's what we think is the main noise for aging.
Interesting.
And we've also, the other cause of aging that we found is cell stress. We damage nerves and they age rapidly. So that's the other issue. There's probably others. Smoking, chemicals, for example, we know accelerates biological age pretty dramatically. But the question is, can you slow that down, or can you reset them to get those ping pong balls to go back to where they originally started in the game? And we think we've found a way to do that.
What can you give me hints? Whose fault is it in the balls not coming back? Is it the proteins themselves? Are they starting again? I've been obsessed with the protein folding problem from the AI perspective. So is it the proteins, or is it something else?
Well, we know who hits the balls and recruits them, so that the break is recognized by proteins who send out a signal through phosphorylation, is a typical way cells talk to other proteins. And that recruits those repair factors, those ping pong balls, to the break. So the cell's actively doing this to try and help itself. But we don't know who's to blame for them not coming back. That could just be a flaw in the quote-unquote design. I don't think that there's something saying, well, 1% of you balls proteins never go back. I just think it's hard to reset a system that's constantly changing. We have in our bodies close to 1 trillion DNA breaks every day. And imagine that over 80 years, what damage that does to our epigenomic information. Now, we know that this is, we never know anything in biology, but we
Right.
have strong evidence that this is true because we can mess with animals. We can create DNA breaks and tickle them with a few breaks, maybe raise it by threefold over background levels of normal breakage. And if we're right, those mice should get old. Mm-hmm. And they do. We can actually, we've created these breaks in a way that's titratable. It's like a rheostat. We can send it to 11. I drove my Tesla here. I'm a big fan of Spinal Tap 2, going to 11. If we go to 11, we can make a mouse old in a matter of months. We prefer to go to a level of about 4, and it gets old in 10 months. But it's definitely old. It's got all of the hallmarks of aging. It's got diseases. Wow. It looks old. Its skin is old. It's got gray hair. But importantly, we can now measure age by looking at the scratches. We can look at the epigenome. We can measure it and use machine learning to give us a number. And those mice are 50% older than normal.
So you can replicate the aging process in a controlled way. You can, in a way that you could accelerate it in a controlled way and measure how much exactly it's aging, and that gives you step one of a two-step process to when you can then figure out, well, how can we reverse this?
And now we're reversing those mice.
Is there a good? I love what you said. I mean, in biology, you really don't know. It's such a beautiful mess. Is there ideas how to do that? Is that on the genetic engineering level? Is it like what can you mess with? Is it going to the trying to discover the backup copies and restoring from them? Like what's if it's possible to convert it into natural language words? What are the ideas here?
What is the observer and how do we contact it?
Yeah, exactly. What's the observer and how do you contact? Or if there's other ideas, how to reverse the balls getting lost process.
Yeah. Well, you can slow it down.
Slow it.
But we found that a reset switch recently. We just published this in the December 2020 issue of Nature. And what we found is that there are 3 embryonic genes that we could put into the adult animal to reset the age of the tissues. And it only takes 4 to 8 weeks to work well. And we can take a blind mouse that's lost its vision due to aging, neurons aren't working well towards the brain, reset those neurons back to a younger age, and now the mice can see again. These 3 genes are famous actually because they're a set of 4 genes discovered by Shinya Yamanaka, who won the Nobel Prize in 2016 for discovering that those 4 genes, when turned on at high levels in adult cells, can generate stem cells. And this is, I think, well known now that we can create stem cells from adult tissue. But what wasn't known is, can you partially take age back without becoming a tumor or generating a stem cell in the eye, which would be a disaster? And the answer is yes. There is a system in the body that can take the age of a cell back to a certain point, but no further, safely, and reset the age. And we're now using that to reset the age of the brain of those mice that we aged prematurely, and they're getting their ability to learn back.
This is really exciting, right? Like, what's the downside of this?
Well, the downside is if you overdo it and you don't get it right, you might cause tumors. But we do it very carefully, and we also know that in the eye it's very safe.
Yeah.
We also injected these. We deliver them by viruses, so we can control where and when they get turned on. And in this paper, we've published that if we put high levels in the mouse, into their veins, throughout the body, they don't get cancer for over a year. So I'm so optimistic that we're going into human studies in less than 2 years from now.